Good morning, everyone. Let's come to the session 2 of your course of molecular biology. Let's discuss about the genetic material and its experiments. So, what made certain nucleic acids like DNA to be called as a genetic material, which has been it's has been passing from generation to generation. So, what we call here is what made a certain nucleic acids to name it as a hereditary material. So learning outcomes, here you will end of the session you will come to know like how do we know that all the genetic informations come from the DNA. That's what. So all the genetic informations, when we are saying why DNA is a hereditary material, we'll come to know. Okay, so way back the Mendel, who was the pioneer and the father of genetics, he published his works in 1865, and just go through the pages of the history. Then the work was lost until 1900, where he worked with the peas extensively. He worked with the peas. There he didn't know about DNA and all the gene. He used to call it factors, right, or the factors which is important in particular trait of a plant, like tall or dwarfs and so on. So till 1865, Mendel was very much involved in working with all the discovering the traits, and he assigned those traits for a genetic material called factors. But what is in physical nature? What are those factors? What a biochemical or what bioentity was important in giving a tallness or dwarfness of the plant, or for that matter the shape of the seed or color of the seed and so on? What was the genetic matter? So again, this question was really pursued in late 1940s and 1960s, and then biotech revolutions started at 1970 and so on. So all it started when way back in 1928, when Frederick Griffith was working with Streptococcus pneumoniae strain, where he said there are some factors which is important, which is being passed from generation to generation, or is being transforming. It's transforming another strain, okay, in such a way that the genetic material from one Streptococcus is being transferred to another Streptococcus, and it is making acquiring the characteristics from where it has transferred. So that is how Frederick Griffith and came up with some genetic material experiment, some of the seminal studies. Then in 1944, McLeod and McCarthy, okay, determined that the DNA is a transformation material. So it was Griffith who knew. He came up with an idea. Okay, some strains are becoming virulent, but in 1944 it was answered by Oswald McLeod and McCarthy, like DNA is actually being transferred. We'll discuss that later. So that's what transforming principle. So what's transforming principle is Griffith. Okay, he conducted an experiment to show that transformation in the living cell is by transforming principle. So if one bacteria is being transformed by another bacteria, so what is happening, how it is being communicated between this bacteria and this bacteria? So what material is coming from this bacteria to this bacteria, so that it is acquiring the characteristics of this bacteria, in the sense from where it has been transformed. So what was that material? Okay, so later it was discovered to be DNA. So it's a simple Griffith experiment, where you all know, like when we had taken smooth strains and rough strains. Smooth strains are ones, the strain which has a coating or cell surface was very much smooth. That's the reason phenotypically he used to say it's smooth. So when he used to inject a rat, so a mouse, he used to see that always the mouse died. Okay, so this kind of bacteria which is phenotypically smooth in the cell surface. So however when smooth actually mutates to the rough strains, he used to see it's alive. That's again thing. So smooth is a virulent strain, R is a R is a rough, it's not virulent. But however, what happened is, when he used smooth and heat killed it, he used to see that the mouse was again alive. That means he is killing all the smooth strains. In the sense, you are not using the smooth strains in such a way. Smooth strains are being killed, so that it is denatured. The complete cell lysis happens. Of course, when the cell lysis happens, again it loses virulence. But what actually was the testing of the times is when he has taken smooth strains and when he combined with the rough strains. So what he found was really intriguing. Like once he injected it, okay, once he injected it along with smooth strain and rough strains, he saw the mouse to be dead. So what's happening when he is using smooth strain and rough strain? The mouse is dead. So when he isolated from the dead mouse and he did some histological study including tissue analysis, he found that live cells were live S strains, living S strains were recovered. So what was happening here is the logic was simple. So what he was seeing is that the smooth cells are transferring the the DNA. Okay, now at this time Griffith didn't know about the DNA. It was MacLeod and McCarthy. So smooth cells, the DNA from the smooth strains, are being transferred to the rough strains in such a way, such that it is having all the DNA in it from the smooth strains, right? And that actually is killing the mouse. So the DNA from the smooth is being transferred and to the rough strains, and R is becoming virulent now because the DNA which is involved in which is killing the mouse is being transferred to the R strain and it is killing the mouse, right? And the R strain has been transformed into S strain. So the genetic material from S has been transferred to the R strain. So this is what the experiment is all about. Then again, Oswald and MacLeod and McCarthy, they have again have done pneumococcal infection where they found this method of transformation, and this transformation they analyzed it too, and they did some chemical analysis and so on, and they found it was actually the DNA. Griffith didn't know about the DNA at that time, so it was MacLeod and McCarthy who came up with an answer where he found that DNA is actually transforming, and DNA is a genetic material here. Yeah, so this is what the same experiment he has done, and he found that R strain or RS strain, R strain which is being transformed by the S strain is actually the DNA. It's the DNA which is being passed from S strain to the R strain. Okay. Then Hershey and Chase experiment, again one of the classical paper and one of the pioneering studies where they found that when you have taken bacteriophage, right? So once the bacteriophage was taken, and bacteriophage actually infects the bacteria or E. coli. So they found that the DNA which is being transferred, okay, they have been transferred from the bacteria virus to the E. coli. Actually, it's the DNA which is being passed from the from the virus. So this is how the basic things came up with, where the DNA was assigned to be a genetic material, right? So then all these Hershey and Chase experiments they have done. Okay, so you see that DNA is being injected into into the bacteria. So there, from this they came to know about the DNA is a genetic material and so on. Okay, I hope you understood the concept of DNA being a genetic material and a brief history of that. Okay, thank you so much.